Well, it's now a real treat for me to be able to introduce the very first Orlo and Carol Clark Distinguished Lecturer in Endocrine Surgery. And we are very appreciative to the AAES Foundation and Orlo and Carol for their generosity in endowing this lecture. The speaker today is someone that I'm very happy to introduce. Professor Jack Gilbert earned his PhD from the University of Nottingham University in the UK in 2002 and went on to receive postdoctoral training at Queen's University in Canada. He has had positions in several institutions, but is currently the director of the Microbiome Center and professor of surgery at the University of Chicago. He's the group leader for microbial ecology at Argonne National Laboratory. He is research associate at the Field Museum of Natural History and scientific fellow of the Marine Biological Laboratory, as well as holding a visiting professorship in surgery at the National University of Singapore. Dr. Gilbert uses molecular analysis to test fundamental hypotheses in microbial ecology. He has authored more than 250 peer-reviewed publications and is really one of the most creative and innovative researchers that I know, and I have to say I'm very honored to have him as a colleague at the University of Chicago and very happy that he could join us today. And so let me welcome up Professor Jack Gilbert, who will speak on thyroid cancer and the microbiome.
All right, my voice is loud. I'm just testing the guys at the back, by the way. I'm on a lavalier, so it makes it a bit easier. Number one, I'm not a surgeon, and I was testing some of the people who were, some of the vendors out there in the room next door earlier about their knowledge in surgery, because I have virtually none. And that might seem a little bit strange, me being up in front of you with virtually no experience in surgery and being a professor of surgery. Nothing seems to make sense anymore. That being said, my training is very strange. I'm an entomologist who got interested in bacteria and then got interested in protein chemistry and somehow fell into medicine. And over the last 7 or 8 years, I've been building up a program of understanding what the role of microbes and bacteria and fungi and viruses in our health and well-being. So, again, nothing makes much sense, and there isn't a lot of work on thyroid cancer and the microbiome, but I'm going to give you an overview of where the research topics that we work on in surgery and microbiome and immunology and in cancer and immunology and the microbiome, where these intersect and the hypotheses that I believe will be important in driving this research area forward over the next few years. So, I am a microbial ecologist who's in a department of surgery, and I think this is prescient of how we are driving and changing our understanding of the role of disease, precision medicine, elements of how we treat our patients in microbial ecosystem dynamics generally, right? We're putting ecology back into health and disease. And I've seen that in a few of the talks this morning. I saw that coming to the fore, especially in the anti-PD-L1 study and in our driving of understanding the role of immunobiology in treating these diseases. I think we're going to see a rapid change over the next decade in how we treat some of these cancers. So, I have a couple of disclaimers. I do have 2 companies that I co-founded, Gusto and the Biocollective, around microbiome therapies and microbial diagnostics. So the microbiome, what is it? It's the sum total of bacteria, fungi, and viruses that inhabit your body, right? They are all over you, in your mouth, on your skin, in your colon, in your gastrointestinal tract, any surface whereby there's a mucosa or an interaction with the outside world, you'll find bacteria associated with it. There are some 35 to 40 trillion bacterial cells in the body, around 1 to 1.2 times the number of cells of human origin in the body. They weigh around 2% of your body mass, so there's quite a lot of them there. You want to get rid of that last few pounds on your diet plan, then take a few antibiotics, and they might disappear. They also have around 7 or 8 million genes, genetic information that's used to encode functions which the body has come to rely on over the last 5 to 10 million years of our ape evolution. And we are now extraordinarily dependent upon the biochemical functions that they provide. In fact, when we see a child without bacteria, and there's only 1 or 2 instances, the boy in the bubble in the 1970s, Paul Simon sang about him, that kind of area where we see a child growing up without a microbiome, we do see significant alterations in the physiology of that child that parallel the work that we see in germ-free animals. So there is an understanding that the microbial community that we have in our body can alter and shape the physiology, the endocrinology, even the neurobiology of our bodies. Right? And so this ecosystem is extremely important. Where do we get it from? We get it from our mothers, mostly. The early phase exposure we get very early in life. For example, here we can see a principal coordinate space analysis. Each dot is a microbial community. The closer the dots are together, the more similar those microbes are. Up at the top, you can see oral microbes in yellow. That's the oral microbiome, the sum total of some 300 different species of bacteria and their relative proportions in the mother's mouth. And they're very closely similar. These are a number of different mothers. Down here on the right, we have in red the mother's vaginal microbiota. And in pink, we have the microbiota of an infant in the first few days of life that was born via vaginal delivery. Right? So you get a really healthy dose of vaginal bacteria when you're born via vaginal delivery. No shock there. Okay? Over here on the right, we have the mother's skin microbiota and a baby's microbiome born via cesarean delivery. And in fact, what we've shown subsequently is that it's the microbiome of the first person that handles you physically that is the source of bacteria that come from the skin and colonize the child. So if you do as I did when my children were born and, strip off your shirt in the middle of the OR and grab the baby and stick it on your chest to get that skin-to-skin contact, which apparently is beneficial, then the baby will get your father's microbiome in the skin as the first initial phase. This is colonization. It's exactly the same as when a volcanic island erupts out of the ocean and is colonized by insects and plant seeds, etc., and soil. The baby comes out of the amniotic sac, is released into this microbial world, and is colonized, and that very first degree of colonization shapes their immune response. Okay? It shapes how their immune system expands and reacts to stimuli from the outside world, and that alteration completely and utterly functionally changes how they're going to respond to the world that they are brought up into. And I'll show that with some work that we've published recently. We also know that the microbiome depends upon our host physiology, upon our genome, upon our immune system, but it also derives through that response a uniqueness. Every single one of you in this room has unique microbiome. You are microbially unique. And that's because the sum total of your interactions with the world, right, of your physical interactions, your microbial interactions, the way your immune system has been altered and stimulated, those interactions, the sum total of them, has led to you having this unique microbial profile. Okay? So I can actually use that for forensic applications, and I actually work with the Department of Justice and the National Institutes of Justice to devise ways of using that in criminal cases, completely unrelated to surgery, but we have a very diverse portfolio of research, so we can actually tell when a burglar has been in your house based on the bacteria they leave behind. And I'll talk a bit about that in a second. So what's happening in the world? Well, we think that by altering our microbial exposure from a rich microbiome outside to one living inside where the microbiome is significantly depleted, and whereby cleaning products and antibiotics lead to a death of the good and bad bacteria, we believe that we may be depleting the microbial diversity of our bodies, removing certain functions which lead us open to atopy, allergies, and a general immune and endocrine dysfunction. We believe that by adding bacteria back into the body, we can actually help to stimulate the immune response and protect children from allergic influence. Okay, that's a nice cartoon. So I'm trying to break you in easy, right? The key thing is, when you go onto clinicaltrials.gov, you see that this idea, while it seems very NPR, tree-hugging, and hippy-dippy, this idea has actually led to a significant proliferation of research paradigm. Okay? We are now seeing 399 ongoing clinical, observational, or interventional trials on clinicaltrials.gov that incorporate, at some degree, a microbiome, right? An analysis of how the microbiome changes in response to something. So, for example, alcohol consumption or smoking, how that affects it. And you notice I always put alcohol consumption next to breast milk. These are, I don't know why, that's strange. Or how the microbiome could be leading to the onset of a particular condition. So, for example, psoriasis or autoimmune disease or allergies or chronic pulmonary disease. All of these elements are part of our research portfolio, whereby we see dysbiosis or a shift in the functionality of the microbiome in our body, altering our immune response, our endocrine response, our neurological response, and shifting our probability of developing that disease state. So this suggests that the microbiome is involved in everything. This isn't hard to understand if you think about it, right? Your body is a highly connected ecosystem of ecosystems, right? It's constantly interacting, it's constantly dynamic, and you change one thing in an ecosystem, you will alter something else in the ecosystem. I change the immune response controls the microbiome. Think of the immune system as a gardener or a national park warden, right? You know, they have to keep the good bacteria alive, they have to keep the bad bacteria from proliferating, they have to actually engage and buoy up some of the bacteria that the body needs in order to survive and be well, okay? So the ecosystem is constantly in a dynamic interface and exchange with the immune response, and that leads to constant changes in how the body responds to that changing environment. That's a lot of change. But that change is important. That changes how we understand this dynamic and alter it functionally. One of the key microbiome therapeutic studies, and many of you might have heard of this, is the use of a fecal microbiome transplant in the treatment of Clostridium difficile, a recurrent C. diff infection, right? This is not a new study. 1,700 years ago, there was a Chinese document that designated something called, there's two direct translations, one's golden soup and one's yellow soup. But the same principle is involved. You take the stool from a healthy and pure child, an infant, right? Has to be pure of spirit and mind in the ancient language, but either way, we think of somebody who hasn't been exposed to disease. And then you take that stool and you put it in a broth and you give it to someone with diarrhea. And you make them drink it. And this was highly effective as a treatment strategy, no joke, for treating diseases of the gastrointestinal tract, right? Based on existing Chinese ethical logic around 1,700 years ago. So this is a new strategy that have been employed, and we now employ it regularly for C. diff transformation. Here on the right, you see vancomycin treatment with either bowel lavage or no bowel lavage, and you get about a 30% success rate in the reduction in C. diff infection status. Whereas if we infuse into the colon via an enema, or now via an oral probiotic, a collection of the bacterial organisms that were present in the gut of a healthy person, literally extracted straight from their stool, we get about an 80 to 94% success rate in the treatment of C. diff. Right? I didn't see anybody showing me statistics like that for any other treatment strategy. We get a significant elevation in the ability to treat a disease. Why? Well, I'll show you a bit about that later, but essentially we're replacing the ecosystem of the gut. The ecosystem is damaged, something has taken over, it's now reached a stable state of disease, and we replace that ecosystem with a healthy, functioning ecosystem, and the body recovers. Right? This is not perverse logic. It makes logical sense. We know that we can treat things as diverse as depression and anxiety by altering the microbial community present in the body. In fact, some of our work with our animal models, this is a elevated plus maze, for example. It's about 1 meter off the ground. Either side of the, on 2 sides of the maze, we have a box, and the other 2 sides, it's an open platform. When we take a mouse, a standard mouse, an SPF mouse or a rat, it tends to stay inside the box, right? It doesn't want to be exposed to the world. A mouse that is exposed usually gets eaten pretty fast, and that's not a good thing for a mouse. So the mouse stays inside the box in our animal model. If we take all the bacteria away from that mouse and make it germ-free, it will tend to spend more time hanging outside the box. I'm not going to anthropomorphize a mouse too much, right? But the essential component here is that by altering the microbiome of this mouse, we can fundamentally change its behavior. And this is a vertebrate animal, right? We've actually shown that people with depression have a significant reduction in the bacteria in their gut that produce gamma-aminobutyric acid. This is a immunosuppressant, right? A nervous system suppressant, neurological signaling suppressant, but still a neurotransmitter. They also have an alteration in the production of 5-hydroxytryptophan, which is a precursor of serotonin. Okay? These are changes in how the microbiome processes or mediates the production of neurotransmitters. When we see a disruption in neurotransmitter production, we see an alteration in behavior in the central nervous system of these animals. So in the animal model here, we've actually started giving these animals a probiotic formulation of bacteria that can produce gamma-aminobutyric acid and bacteria that can produce 5-hydroxytryptophan. And we see an immediate cessation of this aberrant behavioral activity and a return of them living inside the box. So we basically regain the ability of that animal to show a normal behavioral response. We know that microbes can play a role in obesity as well. A colleague of mine used to weigh 385 pounds. I won't say who that colleague was because that would give it away. But he lost 113 pounds. The IRB can't tell me anything for pointing my laser pointer in the wrong direction. He lost 113 pounds by taking a traditional Chinese whole grain medicine diet for 9 months, okay? And he checked his microbiome at the beginning and at the end. And at the end, there was one bacterial taxon, very abruptly, that became, or dropped below the level of detection. Right? It was very abundant at the beginning, and it was virtually absent at the end. This was Enterobacter cloaciae B29. He managed to culture up this particular strain from his stool, and when he gave that to an animal, the animal changed fundamentally its immune profile, its endocrine profile, and also, as you can see, its weight. It put on a hell of a lot more fat tissue for the same calorific intake and the same degree of activity as a mouse without that bacteria inside it. And this was the same for germ-free mice and for SPF mice that had this organism added. Okay? This is a obesity-generating bacterium. And that suggests that obesity could be a transmissible infectious disease if you had one of these organisms associated with it. We've done an awful lot of work with my colleagues Jean Chang and various other colleagues across the country at University of Chicago and elsewhere to try and understand how this happens. What is the role, the mechanism by which microbial dysbiosis or changes in the bacterial activity in the gut can elicit shifts in metabolic activity in the rest of the body? And a lot of it has to do with endocrine disruption. Disruptions in the hormone levels associated with the body that lead to the processing, management, or maintenance of energy production or storage inside the body. And this is because the bacteria produce small molecules, metabolites, which transmit into the bloodstream and alter physiochemistry inside the body, and also change immune responses, causing a pro-inflammatory state across the body. I'll talk a bit more about that in a minute. A paper that we published last year in New England Journal of Medicine focused on a particular element that I think is of relevance here, and one of our colleagues, Ray Grogan, in Department of Surgery at University of Chicago, has been promulgating this idea that alterations in the exposure of people early in life can have effects upon immunology and endocrine physiology at a later stage. However, this is an Amish farm. It's in northern Indiana, right? This farm has a rich and diverse lifestyle associated with it. It's it's an Amish environment whereby the Amish, who come from Central Europe, basically came to America and adopted a lifestyle whereby they maintain a lack of technological advance beyond about the late 1700s. So there's no iPads, no televisions, no electricity, no cameras, no technology allowing them to use the farm environment, so they still use horses to pull the plow, okay? And the children do not have any technology either, and they work very closely with their parents on the farming environment. It's a very work ethic, strong community. I take my children, who currently sat at the pool all day yesterday, to this farm and show them that children can survive without an iPad in their hands for a prolonged period of time. This is a very different environment to what most of us brought up in. But there's an allegory here in the fact that the Hutterite community, who live in North Dakota, they have a very, very similar lifestyle and genotype background, right? They come from Central Europe ancestrally. They have the same biomarkers and genomarkers for asthma as the Amish, nearly identical genotypic risk ratio associated with that disease for asthma. And they live at home a very similar lifestyle to the Amish, right? So no technology, no iPads, no phones, no cameras, etc. But on their farming environment is completely different. They live in big communal farms, the Hutterites, and the Hutterites have these very technologically and mechanistically advanced farming environments which actually sever the likelihood that a child can be exposed to that ecosystem. And what we see is, the children can't go on the farm because it's technologically advanced. It would be against health and safety regulations to allow a child into that environment, right? So the kids get no exposure to the farm on the Hutterite farms. But the Amish, the kids are basically being bombarded with bacteria and allergens from this farming environment from birth, okay? And the two key things here, the Amish have an asthma rate in their population of 4%. The average for the US is around 8 or 9%, and the Hutterites have a rate of about 35%. Right? So a very significant elevation in asthma risk for a population which has the same genetic risk factors as the Amish. Okay? Now, when we look at the immune system response to these children, we see a couple of interesting things. For example, the peripheral blood leukocytes from the Amish children have increased proportions of neutrophils, decrease in eosinophils, and about the same level of monocytes as both the Hutterite kids. The neutrophils, and this is the most interesting thing from our perspective, in the Hutterite children were significantly more aged, i.e. they had been in circulation for longer than in the Amish children. And work we've done in sickle cell disease and vaso-occlusive crises with our colleagues at Northwestern has highlighted that aged neutrophils are significantly pro-inflammatory. So essentially, the Hutterite kids are walking around with inflammation bombs in their aged neutrophils associated with their blood system. So any allergen coming into the environment has a significant chance, and I'm talking about the body's environment, has a significant chance of causing a massive pro-inflammatory reaction and leading to a significant alteration in physiology. Interestingly, there was no difference between Amish children and Hutterite children in Treg populations. So this isn't associated with your standard alteration in the ability of the immune system to dampen down inflammatory responses. This is driven by another process. When we look at the gene expression profiles associated with the leukocytes, we see a significant differentiation between Amish and Hutterite children. And when we break that down into a mapping of the significant relationships between expression profiles associated with markers in the immune system, we see two key components here that stand out like sore thumbs. Number one is TN-alpha, so tumor necrosis factor alpha, and then we see IRF, an IRF3, in fact, or an IF7 show a significant alteration in the response. They are massively upregulated in terms of processes of immune activation in the Amish children. And these are innate immune markers associated with microbial exposure, right? So these children are being exposed to this rich, diverse microbial ecosystem, and it's activating their immune response to become, have a higher turnover associated with the disease association, and therefore leading them to have a significant downregulation in their aged neutrophil population because of constant resurfacing of neutrophils in the blood system. This leads to a reduction in the likelihood of them developing asthma. Also, we hypothesize. To test that hypothesis, we basically took dust, just dust, right, from the bedrooms of children in both populations, Amish kids and Hutterite kids. We suck up dust from their bedrooms, which is difficult because Amish and Hutterite parents are extremely clean. Okay, it's a religious desire to make sure that cleanliness is next to godliness, so they suck up all the dust already before we come around, so we have to look in all the nooks and crannies. But we take the dust from an Amish child's bedroom, and we basically blow it into the nasal cavity, into the nasal mucosal interface of a mouse model that has been sensitized with the valbumin, okay? So essentially you sensitize this mouse with the valbumin. If you then re-expose it to a valbumin later on, it's going to suffer a severe immune response, and you'll have eosinophilia and airway hyperresponsiveness associated with that exposure. When we take the dust from the Amish children's bedrooms and we blow it into the nasal passages of these mice, they show a significant protection against the eosinophilia and the airway hyperresponsiveness. Exactly, that's what I was thinking. It's all that dust in the nose talk, I know. Does that to me every time. But when we take the dust from the Hutterite children's bedrooms, it has no protective effect. Okay, so dust from two different environments seems to have a differential impact upon the likelihood of a mouse developing an immune response that is an analogy towards asthma, okay, in terms of airway hyperresponsiveness. On a follow-on to this, we haven't published this yet, we're working on it right now, but if we take the dust from Hutterite children's bedrooms where there is an asthmatic child or asthmatic parent in the house versus a Hutterite community home where there is no asthma, they are also differentially protective. So again, protection from a no asthma house dust sample and no protection from an asthma house dust sample. So the house in which you grow up in alters your immune phenotype, changing your likelihood of developing that disease. And we are currently trying to isolate the bacteria and the fungal agents from that dust and to try and determine specific probiotics combinations that could be protective in that model. And again, that sounds very hippy-dippy, but if we're right, then the simple administration of a nasally infused probiotic formulation, either dead or alive, early in life, could significantly retard the development of this disease condition in these populations, and maybe populations globally, which is another tool in our arsenal for fighting these diseases. So cancer in the microbiome has an interesting history. We've known about it for years. Obviously, viruses. Obviously, everybody knows that viruses play a role in cancer development and activation. And we started to show that bacteria do as well. There's an interesting analogy here. If you look at cancer population rates in Japanese from Osaka versus Hawaiian Japanese, we see that acculturation into a Western lifestyle from a Japanese lifestyle appears to significantly alter the cancer rates in those populations. So, for example, when the differentiation between Osaka Japanese to Hawaiian Japanese shows an increase in prostate cancer, an increase in colon cancer, decrease in stomach cancer due to a decrease in the rate of things like H. pylori in the stomach cavity, and then an increase in breast cancer. So there is a basic analogy for understanding that when you change your lifestyle or your lifestyle exposure rates, diet, upbringing, environment, etc., you can change your disease rate. And when we look at hypothyroidism, we see a similar kind of analogy. I mean, it is an autoimmune disease. Hashimoto's disease is an autoimmune disease, so we know that hypothyroidism is associated with those kind of components. And most autoimmune diseases have some kind of microbial trigger. That's the work from the asthma studies that we're doing and many other conditions, such as food allergies and pet allergies. Most atopy has some kind of microbial trigger. So we're trying to understand that dysbiosis and how it leads to disease onset. We know that germ-free rats have smaller thyroid glands. An antibiotic treatment can alter thyroid function through changes in lipid polysaccharides, so gram-negative bacterial exposure, and selenium competition, so how microbes alter selenium concentration by either degrading selenium and using it as a metabolic product in the gut and changing its availability to the host. So these kind of components suggest that there is a link between microbial activity in the gut, microbial exposure rates through translocation into the blood system, and alterations in thyroid activity. And thyroid cancer has these similar associations. Essentially, exposure to a microbial-rich environment in childhood seems to reduce the risk of developing papillary thyroid cancer. This is based on this study from Clark et al. Is Christine Clark in the room? No, that's bad. Where are you? That would have been cool. Because I like this paper. But either way, there's a poster in the room next door, because I went and previewed the posters, which seems to suggest that there's population differences in papillary cancer rates associated with these groups, in the fact that lower socioeconomic status may actually be an elevated factor in the development of some of these diseases. If that's true, then we need to embrace that understanding and bring it into research paradigms. We know that mice without bacteria can show increased intestinal adenomas, and this is work that we've done in colon cancer therapy. This original study from 1997 showed that mice with a reduction in intestinal adenomas were generally germ-free, and we have about a twofold reduction in the rate of polyp formation and intestinal adenoma generation from carcinogenesis associated with notobiotic animals. So you remove the bacteria, you remove the likelihood of progressing towards a disease. And we also know that from screening human populations that we see a significant elevation in certain risk factor bacteria associated with colon cancer development. Obviously, colon cancer is an easy one, right? Because it's bacteria, the cancer's in the colon, and so there's bacteria and colon cancer going side by side. But hopefully we can embrace that differentiation. In the fact that Fusobacterium here, Fusobacterium nucleatum to be specific, is significantly elevated in tumor-associated tissues compared to healthy tissues in this population. And when we do fluorescence in situ hybridization, you actually see Fusobacterium nucleatum significantly associated with the tumor burden. The interesting thing is that it differentiates based on a number of factors. Number one is, this is our own work from 2013, differentiates based on density. So the density of Fusobacterium or other cofactors associated with the tumor significantly elevates or reduces the risk of having a polyp trigger into local inflammatory responses, including immune responses associated with tumor infiltration. And so we see a differentiation in terms of carcinogenic development associated with the infiltration of immune responses with cancer, with microbial density associated with the actual tumor. Okay? Then when we look at different responses, take a backtrack. I did just get off a plane from Singapore, so I apologize. When we look at the community profiles associated with different cancer rates, we have a way of differentiating these populations. The first way is to look at the function of the bacteria associated with the cancer or with the gastrointestinal tract or the mucosa. So this tells us what those bugs are doing. And then we can map those bacterial community reads to protein families in the gut and actually try to reconstruct the metabolic pathways of those microbes. Remember, this is a very complex community of microbes, so we have to map and interact how they actually function. And then we can also look at what organisms are there, and this is highly important because it allows us to differentiate your microbes from your microbes and see how their response variables interact. And to do this, we actually reconstruct the genomes of the individual bacteria associated with different samples and then map all of this functionality into those genomes and determine how different strains of a particular bacterial species can influence disease etiology in different contexts. This is a study paper that used this approach. It just came out in 2017, not from our work, but using this same approach to identify the causative microbial influences in colon cancer associated with an Asian population, particularly Chinese, Han Chinese. And what they identified was, again, Fusobacterium nucleatum in this population was significantly elevated in tumor tissue, but also this Parvomonas micra, another organism species never identified in a Caucasian population of colon cancer patients, was significantly elevated. And again, Eubacterium venturosum is an organism which was downregulated in the tumor population. Using network homological theory, we're actually able to, and mathematical theory becomes extremely important in trying to disentangle these components. I myself am a systems mathematical biologist, and so we can use this information to try and identify the particular associations between bacteria and between tumor and cancer outcome factors that alter that disease etiology. This is important, right? If we're looking at a Chinese population, we identified different biomarkers of disease from a Caucasian population. If we may see differences in an African-American population versus a Caucasian population or a Hispanic population, we need to be able to identify how socio and racial factors influence the distribution of this disease burden in the state in terms of identification of biomarkers and potential identification of treatment strategies. Talking of treatment strategies, we heard about anti-IPLD1 from the group at Sydney. It was a really good talk. I really enjoyed that one. And the key thing here is that we started to look at how microbes in the gut and how probiotics can influence that immune state change and could potentially be used as a combinatorial therapeutic in say, imelapsepub activated anti-PD-L1 strategy. So to do this, we try and identify the predictive biomarkers that allow us to direct treatment towards benefit by taking not just the tumor, but again, a fecal sample to look at the microbiome and potentially healthy tissue outside of the tumor environment. By doing this, and we're creating these databases of fecal microbial samples alongside tumor samples, alongside the tumor microbiome, we're starting to understand the differences within our populations and use this for precision medicine. Some work from our group, from Tom Gajewski at University of Chicago and a French group simultaneously published in the same edition of Science in 2015, identified that microbial probiotics can play a dramatically significant role in shaping the efficacy of anti-PD-L1 therapy in a mouse model of melanoma. In this instance, this is the most beautiful thing I love. Pardon me. Should cover that. I get excited. So in the untreated mouse model, you obviously get a significant increase in tumor volume days post-injection. If we just give them bifidobacteria, the BIF there in purple, or the PD-L1, the anti-PD-L1 immune blockade therapy, you actually see a highly similar response variable in the mouse model. Doesn't mean it will work in humans, right? But just giving the mouse a probiotic bacterial organism can significantly reduce tumor volume. Let that sink in. This is a standard probiotic, right? Bifidobacteria is found in every child, every infant, in most populations around the world. And when you put it into the gut, it stimulates the immune response. It stimulates the immune system, actually leading to activation of leading to immune activation and shaping how the immune system responds to alien bodies in the body. And I will show that with surgery in a couple of seconds. But and then when we add anti-PD-L1 plus bifidobacterium, the black line here, we see an incremental increase, significant increase in the efficacy of both as a combinatorial therapy. That's super cool, right? We can add probiotics plus an immune blockade therapy element and lead to a joint immune system response activation, significantly increasing tumor infiltration and therefore tumor clear out. So I think that's very cool. Didn't work. Sorry. So when we look at metastatic melanoma, patients treated with anti-PD-1 checkpoint inhibitors, we've actually started to survey, using our collaboration at Mayo Clinic and Harvard, survey the microbial communities associated with the two components in responders versus non-responders in the patient population. And we see that responding patients have a significant increase in the abundance of ruminococcus. Ruminococcus is involved in immune regulation in the gut, whereas decreased microbial diversity and an increased abundance of Bacteroidealis was observed in non-responders. And Bacteroidealis and the subvariant species thereof can, in certain instances, cause an increase in inflammatory responses in the gut, which can lead to an inflammation-based inappropriate activation. So we need to unravel these connections. We see differences in the microbial community of patients that respond or do not respond. In our animal models, we're able to show that microbial therapeutics and probiotics can influence disease outcome. So if we can try and disassociate and understand and elucidate the significant mechanisms of interaction between those covariables, we could play a very significant role in shaping therapy in humans. Just to finish up now, I want to talk about surgical infections. The immunocentric view of surgical infections in sepsis and critical illness is that essentially microbes initiate disorder, i.e. you get an infection, it causes systemic changes in the body's immune response, the change in transcriptional levels of immune activation. But essentially, if I give the patient antibiotics and I wipe out that infection, I've done everything I can. Then the microbes aren't important anymore. It becomes an immunoinflammatory disorder, and that's the primary driver of organ failure and death. Okay? This is the dogma. And there will be few surgeons around the world which would disagree with it, but it's changing. Okay? The problem is that you can't get rid of the bacteria. When we've done studies where we go into an OR, okay, and we place plates around the room, around the patient, in the air, there are bacteria circulating in the air from every single person in that room. The patient, the doctors, every component, every biologically relevant organism in that room is emitting bacteria at a rate of about 38 million bacterial cells per hour, right? So if that was the case, that microbial infections were being led by microbes being transferred from the doctors, etc., then everybody would be getting sick. But you cannot eradicate, you cannot completely source control your environment to prevent microbial contamination of a wound site or of an infective element. It's just technically impossible. So the body is constantly being bombarded by bacteria. They are omnipresent. And what's important is that the microbes in the body and these infective agents and the body itself are in constant communication. So microbes communicate basically with the host via PAMPs and MAMPs and bile acid conjugation. The host communicates with the bacteria and how the host is treated. So, for example, if you give the host opioids, it can activate bacterial pathogenesis and virulence pathways. That's a really interesting point. Give your patient opioids and it can make them sicker. And the microbes communicate with the microbes via quorum sensing exchange of genetic material. We call this sociomicrobiology, the Facebook for bacteria, evolution of bacterial internet. Understanding that can have a huge effect on how we treat a patient and how we respond to a disease. I've got that, bidirectional molecular communication. So a good example of this is that when we treat an animal with a surgical procedure, for example, we actually cause stress in the body. And this stress leads to changes in microbial phenotype. The particular phenotype or activity of a bacteria can be altered by that stress. So, for example, here we give our animal a hepatectomy, and then we do an intracecal injection. It's a 30% hepatectomy, an intracecal injection of Pseudomonas aeruginosa, and then we wait 24 hours. Okay, the stress is the surgical infection, the surgical activity, the surgical practice. The infective element is Pseudomonas aeruginosa. Wait 24 hours, then we grow up the Pseudomonas. Running out of batteries in this thing. Grow up the Pseudomonas on selected media plates, and within 24 hours we see a smooth versus a wrinkled phenotype appearing on our plates. And you can see down here that the smooth versus wrinkled phenotype show very different rates of surgical infection and mortality in our sham animal model. So we give our animal model a hepatectomy, a surgical stress. It changes the phenotype of the animal, of the bacteria associated with its gut that have caused colonization. And in our hepatectomy model, we see a significant increase in these wrinkle types, which actually cause death in our sham animals when we re-inoculate. These are highly virulent strains. But essentially, Pseudomonas aeruginosa, when it colonizes, isn't virulent. So it's important. Non-virulent strains activate in the body, and the only way that they become pathogenic is when they experience the stress of that environment. Not background immune activation. That's not what's causing death in this model. And it's not the particular bacterial species. It's how the bacterial species are shaped by the stress they experience upon surgery. And another nice video to highlight this. When we do a gastrointestinal resection to remove a cancerous polyp, we do two things to the microbes to stress them. Number one, we allow oxygen to penetrate into the gastrointestinal cavity. The anaerobic bacteria do not like this. It causes stress. And we pump the body full of antibiotics. Again, another stressful event causing double stress. And then we finally, when we join up and we produce an anastomotic junction, the body does one more thing. It starts to suck phosphorus out of the gastrointestinal cavity through our alkaline phosphate activity. That starves the bacteria, and some of them panic, and they start to form a new phenotype, whereby they bind onto the anastomotic site and activate a collagenase phenotype, which starts to break down the collagen in the gastrointestinal wall, causing leak. So what's important is if we do not do the surgical element on here, but we inject the pathogen, there's no activity. You need the stress to turn this bacterial commensal organism into a virulent pathogen. That's essential. And that differentiation, it may be what's leading to the vast majority of surgical infections we see. To track that down, we wanted to see what would happen. So, in this instance, we've had an infection and we've got leak, and now we've got severe sepsis in our patient. Right, so what happens to that sepsis, and how can we maybe change that? We produced this new model of surgical activation of microbial pathogenesis by, number one, doing our standard 30% hepatectomy, and then we intrathecally injected a concoction of 4 dangerous pathogens that we isolated from an ICU patient who passed away. 2 multi-drug resistant strains of marcescens and osteotosa, and then a tetar strains of Enterococcus facialis and Candida albicans, which was a standard fungal pathogen. Those organisms, when we inject them into most animals, intraperitoneally or intrathecally, do cause an infective state, but not necessarily as much as you'd expect, and I'll show that in a minute. Post-operative day 1, we've done this 30% hepatectomy, we've done the intrathecal injection of pathogens. Then on post-operative day 2, we do 2 things. Number 1, we sacrifice some of our animals, and we do blood, liver, lung, and spleen biopsy tissue analysis and culturing, and we do demonstrate that the pathogens translocate out of the gut and are then distributed and infecting organs, so causing system-wide organ-based infection and sepsis, okay? And then we do one thing to start with. Post-operative day 1, we give the animal an enema of either a fecal microbiome transplant, the golden or yellow soup from Chinese law, from another animal, or we take that fecal microbiome transplant and we autoclave it. That's the AC. And then we just let them feed as per normal. So fecal microbiome transplant or autoclave stool. Post-operative day 2, we do the same thing. We repeat that enema, fecal microbiome transplant or autoclave stool, and then we assess mortality up to POD 7. There's 2 things I want to point out. Number 1, the microbial community changes dramatically in the gut of these animals. So in non-treated animals, we have a lot of Bacteroidetes, a lot of lactobacilli, and a rich, diverse microbial community. Post-operative day 1, what we call, where we start to see this collapse of the microbiome, we have an enrichment of Enterococcus, an enrichment of Firmicutes, and a significant reduction in bacterial diversity. And then by post-operative day 3, we've either given the animals a fecal microbiome transplant up on the top or an autoclave stool transplant up on the bottom. And you see in the bottom, after the autoclave stool transplant, we see this emergence of what we call a pathobiome, a massive enrichment of Enterobacteriaceae and Enterococcus, complete collapse of the ecosystem, the diversity gone, okay? And this ecosystem is now highly inflammatory, highly pathogenic. The fecal microbiome transplant, however, on the top, seems to rescue or restore the core microbiome that was present in the non-treated animals. So you get an increase in Bacteroidetes, an increase in lactobacilli, an essential restoration of their health. Looking at mortality for these different components, hepatectomy alone, without injection of pathogens, 100% survival. right? Our surgical residents are good at what they do, okay? Hepatectomy plus the pathogens plus a fecal microbiome transplant allows a 90% survival rate for our animals, okay? If we do the hepatectomy plus pathogens plus autoclave stool, near 90% mortality, which is the same as hepatectomy plus pathogens. So a fecal microbiome transplant rescues these animals from sepsis. That's pretty cool, I think. And that allows us to have a new strategy. Immune stimulation via fecal microbiome transplant can lead to systemic shifts in how our body's immune system activates and clears out these foreign bodies, whether they be a cancer or a dangerous pathogen associated with body organs. We can see that the immune system response in various distal organs is significantly altered. So this is liver, spleen, and cecal wall. On the non-treated animals, that's the baseline, so it's hence all black. Seeing significant alteration by POD1, that's the defined septic state. And then in POD2, with an autoclave stool, we see a massive activation of immune responses and inflammatory responses associated with these distal organ immune systems. Whereas in postoperative day 2 with the FMT in blue, we see a significant quietening down of that inflammatory response. So it's working, and it's working not just in the gut, but distal to the gut, in the organs, in the whole body. The immune system allows for communication throughout the body's environment. Interestingly, we also did an intraperitoneal injection of these 4 pathogen communities. So basically going for broke and seeing if we cause massive septic infection via intraperitoneal activity, can we rescue them? And indeed, we can get complete or near total clearance of these healthcare-associated pathogens from distal organs and the gut via the IP-injected mice following an FMT produced through a rectal enema. That's cool. So we can it's not just an intracecal injection of these pathogens that can be cleared out following translocation out of the gut into the organs, but even if we inject it intraperitoneally, we can clear out the infection with an FMT. And we've actually defined a collection of 12 organisms from these mice that when cultured up and put into a defined probiotic has exactly the same effect. And that's the work that's actually being spun out as a company right now. So I've finished there. Shameless plug, I have a book coming out on June 6th. So if any of you have a child coming into your family, grandchild, or a child, or you're expecting children, then you can buy this, and it explores some of the relevant science behind those elements. Myself and my colleague Rob Knight down at UCSD wrote this with Sandra Blakesley. I should have put that down as a disclosure plug. Anyway, it's a book, right? If you want to buy it, buy it. If you don't. If you want to buy it and you want to send me a copy, I'll sign it. There we are. And I just wanted to highlight the Microbiome Center. This is our multidisciplinary initiative, everything from human biology and animal biology all the way through to marine and terrestrial and soil ecology. And we actually combine the expertise from these very disparate groups of scientists who don't normally communicate with each other, and we allow for significant acceleration in the scientific dogma in a particular discipline by combining those expertise components. So we're really excited about that. And I'll leave it there. Thank you very much.
Jack, thank you very much. That was wonderful, very inspiring. The only problem is you're such a good speaker that it's going to really look bad for me shortly when I have to give my presidential address. But be that as it may, I'd like to invite Jeff Thompson, who's the head of the AAES Foundation, up. And Orlo and Carol, if you could come up, please.
Might be tricky. We can still activate this. Thanks.
Thank you. I just want to tell you a little bit about the history behind this lectureship. Orlo and Carol have been such an integral part of the AAES since its inception. And last year at the foundation meeting, board meeting, we decided that we wanted to do something to honor Orlo and Carol for their years of service to the AAES. And we came up with this idea of a lectureship, and this lectureship is being supported entirely by the AAES itself as well as the foundation. And we're very proud that Dr. Gilbert is the first lecturer, gave an outstanding presentation this morning. I'd like to just ask Orlo and Carol to present this to Dr. Gilbert. And Orlo and Carol, we look forward to you being here every year for many years to come, and we thank you for all that you've done for the AAES. Okay. And we also have a AAES tie for you. Yes, yeah, you should wear a tie, as well as your honorarium from the AAES Foundation. Thank you so much.
Thank you. So we're going to now take a break until 11. So please be sure to get lots of coffee.